Hello first year medicos, I am Dr. Poonam Agarwal, Professor Biochemistry. You'll be shortly appearing for university examination in biochemistry where many topics you have to revise in a very short time. So I just thought of helping you in certain series of lectures on YouTube, how to revise various topics. So I'll be making small videos which will help you to revise the topic fast. It will give you a boost in your preparation. So let's start with the topic today which I have selected. The topic for today will be the lipid metabolism because it's a very lengthy metabolism. So I will be dividing my session in two parts. Today I'll be recording the first part where I'll be guiding you how the lipid metabolism need to be revised. So in lipid metabolism, there are many topics which you need to revise before appearing for phase 1 MBBS 1 university examination, various universities pan India. So the topics which are there in the lipid metabolisms are as follow. Number 1 is the fatty acid metabolism, where we have two aspects of it. One is the synthesis of fatty acid and another is oxidation of fatty acid. The oxidation of fatty acid, the major oxidation which we talk is the beta oxidation, but we also have alpha oxidation and omega oxidation of fatty acid. So today's topic is fatty acid metabolism, which I'll be guiding you how to revise it completely. Obviously in this short video I'll not be discussing each and every aspect of fatty acid metabolism, but overall picture I'll be giving you, which will help clearing your concept of this metabolism and will give you confidence in appearing for the university examination. So let's start with the first of all I will discuss you the synthesis of fatty acid, the core aspect, the key points about synthesis of fatty acid. Synthesis of fatty acid, see, it's a long question for you and also a short answer question for you. And nowadays they have started giving you multiple choice question and case-based questions. So this topic is important there also. So you need to know this topic completely. So I'll be just telling you the core aspect of this pathway. When we talk about the synthesis of fatty acid, the human cell is able to synthesize the fatty acid by a process called fatty acid synthesis, and the whole process of fatty acid synthesis is actually taking on an enzyme complex which is known as fatty acid synthase complex. You can call it as FAS complex. FAS complex. Why I'm telling you this FAS complex? Because I have seen many of the university paper, they have given you this terminology FAS complex as a short note for you. So FAS complex is the site, it's an enzyme complex which is actually acting as the active site for the synthesis of fatty acid. Synthesis of fatty acid is a cyclical process, and the whole cyclical process is actually taking place at the active site of this FAS complex. FAS complex, it's a cytosolic enzyme complex having 2 units of enzyme. So it's a dimer, and 2 units of enzyme is arranged in an anti-parallel fashion. This is first unit of enzyme, the second unit of enzyme. They are arranged in an anti-parallel fashion. You need to know there are 7 catalytic domains on each of these unit of FAS complex. And this 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7 catalytic domains are there. These 2 enzymes are same, so it's a homodimer. We call it as homodimer. FAS complex is a homodimer where 2 units of enzymes are exactly same. The only catchy point over here, you should remember that these 2 enzyme units are not arranged parallelly, rather they are arranged anti-parallelly. If we talk about this domain, which is cysteine SH, this area will be cysteine SH, and this is the another pentathene acid site, which is going to be a part and parcel of the active site. So this is how FAS complex is arranged, and you're supposed to know the catalytic domains which are present on each of these enzyme units. And there are 7 catalytic domains. You need to know their names also. So you have to have the familiarity with all the domains of this FAS complex. If you want me to write all the 7 catalytic domain, I can tell you there are 3, 4, 5, 6 and 7 catalytic domain. First catalytic domain you can write is the acyl transacylase, Then the second one is going to be malonyl transacylase. Then you have the third catalytic domain as ketoacyl synthase. Then you have ketoacyl reductase domain. Then you have hydratase domain, which is going to remove the water as to create you the enoyl chain, or the chain having double bond. Then you have enoyl reductase, and then you have final catalytic domain, which is thioesterase, which is going to finally cleave out the product, that is palmitic acid. Yes, you should remember that human is able to produce you 16 carbon saturated fatty acid, that is 16:0, and the name for this fatty acid is the palmitic acid. So by this process of fatty acid synthesis on fast complex, human cell is capable of producing you only palmitic acid. Shorter and longer chain fatty acids are not produced on this fast complex. That's something very important you should remember. And for making this 16 carbon palmitic acid, you need to have 7 cyclical reactions. I said it's a cyclical process, means the same process is repeated again and again on these active sites, and you need to have 7 cyclical process for making 16 carbon palmitic acid, which is going to be the final end product, and that will be seen like a tail at pentatene site. So you know there are 2 catalytic domains on the fast complex, and these 2 catalytic domains, 2 active sites, not catalytic domains, rather the active sites. This highlighted portion is the active site. So 2 active sites are simultaneously involved in synthesis of fatty acid. So at once we tend to have a 2 fatty acid chain on this fast complex after completion of 7 cyclical reactions. Each active site will have 7 cyclical reactions. So at once we'll have 2 fatty acid chain getting produced on this fatty acid synthase complex. And I'm not going in detail of those cyclical reactions, but overall picture you should know that whenever we talk about synthesis of fatty acid, we need to have this important complex that's called fast complex, which is having 2 units of enzyme, and each unit of enzyme is having 7 catalytic domain, and they are arranged such that you tend to have 2 active site, and each active site is involved in the process of cyclical fatty acid synthesis. And after 7 cyclical reactions, which will require these following enzymatic domains of fatty acid synthase enzyme, you need to have these catalytic domains in each cycle. Acyl transacylase will be required only in the first cycle, and thioesterase will be required only in the last cycle to release the final product. But other remaining domains of fatty acid synthase chain will be required in each and every cycle during the synthesis of fatty acid. But when we talk about the synthesis of fatty acid on this fast complex, we need to know what precursor is required. So as far as the precursor for fatty acid synthesis is required, you should know that whenever we talk about first cycle, we have acetyl-CoA, and second precursor is going to be malonyl-CoA. We need to have 2 such precursor to start the fatty acid synthesis. For first cycle, we need to have these 2 precursor. The acetyl-CoA is directly derived from carbohydrate metabolism, and malonyl-CoA is a 3 carbon moiety, and you should know that this malonyl-CoA is actually derived from acetyl-CoA after its carboxylation, right? Acetyl-CoA undergo carboxylation to give you this malonyl-CoA. So in first cycle, we need to have 1 acetyl-CoA and 1 malonyl-CoA. What is the origin of malonyl-CoA? It's a acetyl-CoA only, and the acetyl-CoA which is carboxylated in presence of carboxylase enzyme, that's called acetyl-CoA carboxylase enzyme. That enzyme is going to carboxylate it and going to produce your malonyl-CoA. And once this is produced, there is a lot of stock of malonyl-CoA in the cytosol. Then we can think of synthesis of fatty acid, and in first cycle we need to have 1 malonyl-CoA and 1 acetyl-CoA, raw acetyl-CoA as such. and that will start the process, and we will get the 4-carbon acyl chain at the end of a cycle. But when I talk to you about second cycle, the precursor is going to be only malonyl-CoA. You do not require acetyl-CoA anymore immediately after first cycle. You just need to have malonyl-CoA in the second cycle. Again, in third cycle, you will need 1 molecule of malonyl-CoA. So all cycles will require malonyl-CoA. So 7 cycle means you will require 7 molecule of malonyl-CoA. And if anyone asks you how many number of fresh acetyl-CoA is required, which is not yet modified to malonyl-CoA, you will say only 1. So 1 acetyl-CoA and 7 malonyl-CoA will be required to complete the process, to prepare you 1 chain of 16 carbon fatty acid, that is palmitic acid. So you should know the detail that how the acetyl-CoA is being made available in the cytosol, and then how the acetyl-CoA carboxylase is modifying it to the malonyl-CoA. Please understand, this acetyl-CoA carboxylase is acting as a rate limiting enzyme for fatty acid synthesis, and this is under strict control of hormones and various allosteric regulators. The free fatty acid chain itself is acting as a negative regulator for this acetyl-CoA carboxylase, because it's quite obvious that if already you have fatty acyl chain in the system, in the cell, you will not have fresh synthesis of fatty acid, because anyway the synthesis of fatty acid is a energy consuming process. We need to have NADPH for the process of fatty acid synthesis. NADPH is the source of energy in the fatty acid synthesis. And during this conversion of acetyl to malonyl also we require ATP. So in a nutshell, if you synthesize something, you obviously need to spend energy. So fatty acid synthesis will be taking place only when it is required by the system, when already we have the acyl chain that will act as an inhibitor of this enzyme. That this is something very important. And in addition to this control, you have various other controls also for this particular enzyme. Like for example, you need to have insulin for action of this enzyme, because the monomers of this enzyme is going to be active in dephosphorylated state. So for that you need to have insulin. So insulin is an anabolic hormone. We say insulin is going to synthesize fatty acid. So this is exactly the place where the insulin is going to have its impact. Insulin is going to activate this enzyme by dephosphorylation. So what I have told you till now, that if you're talking about synthesis of fatty acid, you should remember it's a cyclical process, and human synthesizes 16 carbon fatty acid chain, and that chain synthesis actually occurs on fast complex, which itself is a short note for you, and you have to describe fast complex and the active site, the domains of fast complex, and then you have to talk about precursors. The first cycle precursor are these two. The second cycle onward we require only malonyl-CoA, and after 7 cyclical reactions we will have 16 carbon fatty acid chain at the pentathene site of fast complex, from where it is getting cleaved out by action of thioesterase enzyme. So this is in a nutshell the synthesis of fatty acid. And if you talk about further elongation of fatty acid chain or introduction of double bond in the fatty acid chain, they are all separate process which is occurring in endoplasmic reticulum, right? तो ये सब आप पढ़ोगे. So this is just overview. This lecture is meant to give you overall concept of these metabolisms. And of course, wherever you're finding it difficult, or you understand that you have lacuna in the knowledge, you always have to refer your textbook, whatever you have read throughout the year, right? Now, let me discuss few points regarding oxidation of the fatty acid. The major oxidation which we talk about during fatty acid oxidation is the beta oxidation. This is the one oxidation which all the students are familiar with, and but in addition you should know we also have alpha oxidation of fatty acid and omega oxidation of fatty acid. Please understand, the oxidation of the fatty acid which is meant to produce you ATP is the beta oxidation, and when you talk about alpha or omega oxidation, there is no ATP production at all, absolute no ATP production. The purpose of alpha oxidation is different, bachcho. Purpose of alpha oxidation is to oxidize branch chain fatty acids so that they can be assimilated in our system. And purpose of omega oxidation is to oxidize medium chain fatty acid in brain parenchymal cell, which results in the formation of dicarboxylic acid. The exact role of dicarboxylic acid is yet not clear, but this omega oxidation, one thing is for sure, that it is not going to produce you ATP. So alpha oxidation and omega oxidation also you prepare for the short note. And one important disease which is related with alpha oxidation is the Refsum's disease. So this is a clinical correlation of alpha oxidation, where we talk about the defect in alpha oxidation resulting in Refsum's disease. This is a neurological disease which is having the manifestation of difficult vision, especially in the night, followed by retinitis pigmentosa, then cerebral features. So it's a kind of neurological disease which is lethal, at 7 to 8 years of age. And the patient may present with extra phalanges in the lower foot, and the dry skin will be there in the Refsum's disease. So Refsum's disease is one disease which you should prepare for the short note, and that's due to defect in alpha oxidation, where phytanyl CoA hydroxylase enzyme is deficient. So that results in accumulation of branch chain fatty acid. Right, and omega oxidation is we call it as a minor oxidation, because as such biologically it is not playing a very important role. And the exact purpose of production of dicarboxylic acid is not yet clear, but of course if there is some kind of defect in the beta oxidation process, then it is seen that this omega oxidation results in formation of dicarboxylic acid which comes out in urine, and detection of that dicarboxylic acid in urine hints towards the underlying defect in the beta oxidation process. Now I detail you this beta oxidation, because this again is going to be a very important topic for your examination, and the core points regarding this beta oxidation I'll be mentioning you now. This fatty acid beta oxidation is a mitochondrial matrix process. One thing you should know very clearly, that mitochondrial matrix is having all the enzymes for beta oxidation. And just like synthesis of fatty acid, where we saw it's a cyclical process, and each end of the cycle the chain was elongated by 2 carbon. When I talk to you about beta oxidation, this is also a completely cyclical process, and for completely beta oxidizing 16 carbon chain, for example 16 carbon fatty acid chain, complete beta oxidation requires 7 cycle of beta oxidation, right? So you should learn them together. Like during synthesis, the 2 carbon chain, 2 carbon is added in the elongating chain, and you need to have 7 cycle for making your 16 carbon fatty acid chain, and NADPH is utilized during those cyclical reactions. I have told you the enzymatic domains of fast complex which are required for synthesis of fatty acid. Now I'm talking to you a completely opposite thing, and I'm talking about beta oxidation, when the fatty acid chain is cleaved. 2 carbon is cleaved at a time, and you need to have 7 cyclical reactions to completely convert 16 carbon chain into 8 molecule of 2 carbon acyl CoA. Right, and the acyl CoA it enters in the TCA cycle, gives you 10 ATP. So when we talk about production of 8 molecule of acyl CoA, we can easily calculate that 8 into 10, 80 ATP are going to get generated directly in the TCA cycle from these acyl CoA, which are produced from 16 carbon fatty acid chain. And this is not the end. We have more production of ATP from the NADH and FADH2, which we derive during the cyclical process of the beta oxidation. See, each cycle produces you 1 FADH2 and 1 NADH. So 7 cycle produces you 7 FADH and FADH2 and 7 NADH. That again gives you ATP in electron transport chain. So 7 NADH, 7 into 2.5, that's going to be 17.5. and 7 into 1.5, that's going to be 10.5. So this is going to be 28. So 80 plus 28 is going to be 108 number of ATP which is generated from 16 carbon complete beta oxidation. And please understand, with the fatty acyl CoA, when it is there in the matrix of the mitochondria, it is capable of generating you 108 number of ATP in total. And during transportation of the fatty acid across the mitochondrial membrane, this is mitochondrial matrix. This is intermembranous space. This is outer membrane mitochondria. This is inner membrane mitochondria. See, the fatty acid as such is not able to get its access to the mitochondrial matrix. Whenever we are talking about free fatty acid, it as such cannot go to the mitochondrial matrix. It involves a complex shuttle mechanism which is first going to convert it to acyl CoA, at outer membrane towards cytosolic site. This acyl CoA enters the space, and there it undergoes certain modifications. So for all this system, you need to have 4 kind of arrangements, 2 at outer and 2 at inner membrane. These 4 kind of arrangements collectively constitute your Carnitine shuttle. So before we start talking about the beta oxidation which is occurring in the mitochondrial matrix, we need to explain a few points regarding the Carnitine shuttle, that it's a process which is converting the free fatty acid first of all to acyl CoA, right? Okay, so free fatty acid is converted to acyl CoA which enters the space, and there with the help of CPT1 enzyme, this acyl CoA is converted to acyl carnitine. Acyl carnitine. This acyl carnitine enters the mitochondrial matrix by CAT, that's acid translocase system, antipode system, and this acyl carnitine in turn is converted to acyl CoA by 4th arrangement, that's called CPT2. I'm not going in a very clear detail of Carnitine shuttle, but I'm giving you the illustrative example that how the fatty acyl CoA is able to make its entry in the mitochondrial matrix. And please understand, in this Carnitine shuttle, 1 ATP is spent, but that is utilized in such a way that its 2-terminal phosphate is utilized. So we count it as expenditure of 2 ATP. So when we talk about the production of ATP from beta oxidation, total number which is produced is 108, but net production we count it as 106 ATP alone, because 2 ATP is spent during Carnitine shuttle to facilitate the entry of fatty acid into the mitochondrial matrix. And you can very clearly see that free fatty acid is no more free fatty acid by the time it reaches the mitochondrial matrix. Its configuration is changed. Now it's esterified to acyl CoA, and that's quite acceptable to the beta oxidation machinery, because the enzyme which is going to act in the cyclical reaction need the coenzyme A form. So there is no problem if the free fatty acid is converted to acyl CoA, and then it is going to make its entry into the mitochondrial matrix. So whenever you are being asked about the beta oxidation, you should have the overview of the complete knowledge, conceptual knowledge of this beta oxidation process. That is a process which is occurring in mitochondrial matrix, but for fatty acid to reach there, you need to have Carnitine shuttle, which is going to spend 1 ATP and 2 high energy phosphate. And in the mitochondrial process, by a cyclical process which is involving 7 cycles as such, you will have production of 8 molecule of acyl CoA and 7 molecule of FADH2 and 7 molecule of NADH. That's going to give you in total 108 ATP. And because 2 is spent in Carnitine shuttle, the net production from 1 chain of fatty acid, 16 carbon fatty acid, we will have production of 106 ATP. Now, one thing you should know, that when this beta oxidation is going to take place. See, slight amount of beta oxidation is even taking place in well-fed state. When you talk about skeletal muscle, adipose and heart muscle, they oxidize the fatty acid even in well-fed state. But whenever you are being asked about the liver cell and the beta oxidation, please remember the beta oxidation in the liver is going to take place in substantial amount during starvation and during diabetes. These 2 conditions are characterized by lack of insulin, and during lack of insulin, we will have a lot of fatty acid in the blood. which has been mobilized from triacylglycerol of adipose cell. The enzyme, you should know the enzyme which is going to cleave the triacylglycerol of adipose cell and will mobilize the free fatty acid to the blood. That enzyme is named as hormone sensitive lipase. This hormone sensitive lipase is going to be active in lack of insulin, that is starvation and diabetes. Because hormone sensitive lipase is the enzyme which is going to be active in phosphorylated form, and in lack of insulin this enzyme will be in phosphorylated form. So if anyone asks you the source of free fatty acid which is undergoing beta oxidation in liver during starvation and diabetes, your answer should be triacylglycerol of adipose cell, which has mobilized its fatty acid in presence of action of hormone sensitive lipase enzyme. And this free fatty acid, which is circulating so much in amount in the blood, then tend to enter the liver, and when it enters the cytosol via carnitine shuttle, as I have just mentioned, it enters the mitochondrial matrix, undergo beta oxidation, produces a lot of ATP, and this ATP is utilized for many things, for process of gluconeogenesis, for providing the fuel to the cell, right? So this is about the beta oxidation, and this you should know, and please go in detail of cyclic